Hello. So it's 4:15, so I think we can start, yeah? Okay. My name is Natalia. I am a master's student at the University of Manitoba, and the topic of my presentation is molecular biogeography of the high elevation Andean Buckeye butterfly Junonia vestina. I study butterflies of Junonia genus because they have remarkable ability to spread and diversify almost all over the world. Butterflies of Junonia genus originated in Africa in Old World 27 million years ago, and they could diversify in Old World, currently unknown 28 species, and they could cross the ocean to get to New World 4 million years ago to get start for new 18 species in New World. Different ecological niches, new habitats in interaction with lateral transfer genes influence the growth of speciation in New World. The New World Junonia butterflies can be seen from the Canada to the pampas of Argentina, and Junonia butterflies can share 3 mitochondrial haplotype groups. It's A, B, and C, and Central and North America have haplotype B mostly. In South America, haplotype A1 is common. It's around 85%. Haplotype B is less common. It's around 15%. And haplotype C can be seen in just only one species in all whole New World. As I said, butterflies can share mitochondrial haplotypes between species, but they also can share mitochondrial haplotypes within species. So you can see at this slide that, for example, individuals from Junonia vestina can have haplotype C, and other individuals from the same species, Junonia vestina, can have a haplotype A1, and the third one can have haplotype A2. So it's like the same species, different individuals, different mitochondrial haplotype groups. So among all of the butterflies in New World, there is only just one species, Junonia vestina, the high elevated butterfly, which occurred in the Andes of South America. And they can share 3 mitochondrial haplotype groups A, B, and C. Haplotype C doesn't cluster with any other haplotype in New World, but instead it closely related to mitochondrial sequencing of Junonia vilida from Indo-Pacific region. So it's a good illustration of long-distance distribution and gene flow. As I said, like Junonia vestina can be seen in the Andes of South America, including countries such as Bolivia, Ecuador, Peru, Chile, Argentina, so I analyzed 66 samples of Junonia vestina from South America, from museum and personal collection, and DNA was isolated from blacks by using QIAGEN blood and tissue kit. To quantify DNA, we used nanodrop spectrophotometer and qubit fluorometer, and to visualize PCR products and restriction digest, we used QIAGEN electrophoresis device. Genotyping was conducted by using restriction enzymes in specific sites which are diagnostic for haplotypes. And in 3 genes, cytochrome oxidase 1, 2, and 3, to determine to assign each individual to particular haplotype, we designed PCR primers on either side of restriction digest cut sites, which are diagnostic for haplotype. And so we amplify with design PCR primers. We could digest PCR products in order to determine whether these diagnostic cut sites presented in each individual. So we've got the pattern of cuts. We could sort specimens by mitochondrial haplotypes. So at this slide, you can see, for example, haplotype C will be cut at this site and will not be cut at any other site presented in this slide. Also, we had some hardships with museum specimens because it was difficult to identify them by restriction digest, and we used Sankar sequencing of cytochrome oxidase 1. before this study, there were only two mitochondrial haplotype groups known. It's A1 and C, but and also haplotype C was mistakenly identified as haplotype A1 because of based on the short fragment PCR, like short fragment sequencing. But after obtaining the whole entire mitochondria, it reveals that they are different. So, as I said, I analyzed 66 samples, and we found out that 29% of individuals of Junonia vestina have haplotype A1, mostly in central and northern part in South America. Around 15% individuals have haplotype A2, mostly north. Haplotype B can be seen through the Andes of South America, and haplotype C, it's around 27% can be seen central and in central part of distribution. We figure out that it is not any more restricted to Peru as it was, because we could find that in a population from also Argentina, Bolivia, and Chile. We always use short like PCR fragments and small markers to identify haplotypes, but when we have good DNA quality of DNA from specimens, because we're always working with museum specimens, but when we have this good quality, we always try to have next generation sequencing libraries to assemble the entire mitochondria to compare it with other species which are in database, and so we can compare them like more clearly. We can compare what is different, what is it. It gave us like better understanding, better picture, because we could understand a whole mitochondria, not just piece. So as you can see this phylogenetic tree, how it's clustered like haplotype A2, haplotype C, haplotype A1, haplotype B, like it's clustered. So our working hypothesis is to understand where alleles came from, and we know that there are in South America populations, species which have haplotype A1, and we think that Junonia vestina transfer haplotype C to this species, and they instead transfer haplotype B and A2 to Junonia vestina, of course, by hybridization. And again, about the haplotype C, as I told, haplotype C is like of Junonia vestina is closely related to Junonia viridis from Indo-Pacific region. So Junonia viridis crossed the Pacific Ocean to get to New World and pass haplotype C to Junonia vestina. So we are working on our hypothesis, and we look forward to testing this element in our future experiments. I want to thank you, my supervisor, Dr. Jeffrey Marcus, and my lab teams. And also, I want to thank all of you for your attention. Thank you. All good? Yeah? No questions? Okay. No? Okay. Thank you for your attention.
So this is, I think, my best/worst biology joke. Does anyone know how the species Orcinius orca got its common name? So there were these taxonomists and ecologists and marine biologists all on the beach trying to figure out what to name this organism, and then a surfer walked by and said, "Whoa, killer whale, dude." Thank you for laughing. You can see me at the coffee break for some bad jokes about frogs. Great. So hello, everyone. My name is Madeleine Robitaille, and I have just recently finished my master's degree at Queen's University, which is a few hours away from here, so just a little bit down the road. And I'm very excited to be sharing my results today on the contact zone of two deeply diverged frog mitochondrial lineages in southern Ontario. So contact zones are areas where closely related species or lineages meet and sometimes overlap. Contact zones are common in nature and occur across a wide range of species. Pictured are just 4 examples of contact zones in animal and plant life. As we can see from these images, it can be very challenging using only morphological features to differentiate between species at a contact zone. So using molecular techniques, as I will talk about further, might be the only reliable way to differentiate between species at a contact zone. Contact zones come in all shapes and sizes. Pictured are 2 examples. So on the left is a contact zone of 2 Bombina species, and on the right is a Canadian example of 2 pine species. And we can see the contact zones in these examples, and we can see that contact zones can be narrow, they can be wide, they can be linear, they can be jagged, and the size and the shape of contact zones can have interesting ecological implications in terms of the amount of overlap, in terms of competition or ecological interactions between the species. So understanding contact zone dynamics is very important in some species. Contact zones can act as natural laboratories that allow us to study parts of the ecology and evolutionary biology that we don't see elsewhere. Contact zones can have fitness implications. We might see ecological or reproductive character displacement, interspecific competition, hybridization, and all of its concomitant fitness implications. Contact zones are also important for how we estimate and how we classify range estimates and population sizes. And contact zones can be really challenging, so understanding them is really important with respect to conservation policy and management. So how do we classify the species at the contact zone? Now, to give an introduction to the organisms that I study, I'm studying chorus frogs, which are the genus Pseudacris. This is a genus of chorus frogs that occurs in North America. They're very small, spring breeding. In my opinion, they're very cute. In Ontario and Quebec, there are 2 mitochondrial lineages of trilling chorus frogs, Pseudacris maculata, the boreal chorus frog, and Pseudacris triseriata, the western chorus frog. They diverged approximately 10 million years ago and have presumably been evolving in allopatry ever since. The evolutionary history of chorus frogs is a little bit complicated, and so understanding some parts of the taxonomy can cause some challenges. The distribution of chorus frogs in Ontario and Quebec is another issue that has some outstanding questions. Formally, all chorus frogs in Ontario and Quebec are legally classified as Pseudacris triseriata, regardless of the mitochondrial lineage. However, we do recognize that there are 2 separate mitochondrial lineages, which is reflected in the designatable units. Designatable units are a legal term that we use in Canada. It refers to a type of conservation unit referring to populations below the species level. So as we can see here in gray, this is the Great Lakes St. Lawrence designatable unit. This is the designatable unit that would reflect a chorus frog that you would find in Montreal, for example. And this designatable unit broadly maps to the distribution of the Pseudacris maculata mitotype. This one is classified as threatened in Canada. And in red is the distribution of the Carolinian designatable unit, which broadly maps to the distribution of the Pseudacris triseriata mitotype. Now, these distributions are based on results from Laymon et al. In 2007. So we see Lake Ontario here, Pseudacris maculata in red, and Pseudacris triseriata in blue, with the contact zone a little bit west of Lake Ontario. However, this is not the full story because in 2020, Lockheed et al. Found evidence of the Pseudacris triseriata mitotype further north and further east than was previously expected, so a little bit north of Lake Ontario between Toronto and Georgian Bay. This might not seem like that big of a difference, It's only approximately 150 kilometers. However, for an organism like a chorus frog that only disperses approximately 200 meters each generation, this is quite a substantial difference. It is highly unlikely for an organism like a chorus frog that this is due to movement, or at least to movement alone, and it's more likely that it's discrepancies in sampling effort that is causing these differences. So it might be a contact zone that's potentially more broad and more jagged than previously thought. This leads me into the goal of my research, which is to map the location of this contact zone between these two mitochondrial lineages in southern Ontario using environmental DNA. I also did some analyses to test whether there are any landscape or bioclimatic variables that correlate with the location of one mitotype or the other. I'm not going to get too much into these analyses now. For today's talk, I'm going to focus on the location of the contact zone. So I use a technique called environmental DNA to collect my samples. Organisms are constantly shedding DNA into the environment, and we can detect the signature of a species through a whole range of molecular techniques without directly interacting with the organism itself. Pictured on the left, I'm collecting an environmental sample, and on the right, I'm filtering that sample through a peristaltic pump, which collects the DNA on a membrane. There are many advantages of using environmental DNA compared to traditional methods. For one, it's relatively non-invasive and doesn't cause a lot of harm to the organisms. So, like I mentioned, one of these populations is listed as threatened, so reducing harm to the organisms where and whenever we can is important. Additionally, using eDNA is more efficient in terms of sampling effort compared to tissue samples. It also facilitates the study of multiple species, or multiple populations, because we get a more proportional sample of the entire community, whereas with tissue samples, we are restricted to the organisms that we're able to catch, which means that we might be missing some diversity. Additionally, using eDNA facilitates the study of rare or cryptic lineages. Chorus frogs do vocalize, but there are many species that don't vocalize, and so using eDNA can let us detect the signature of these species. There are a few considerations with environmental DNA that we should be cognizant of. It is quite sensitive to contamination, meaning that we do need to be careful with our sterilization as well as with our negative controls. And it occurs in very low concentrations, so we do need to be careful with using sensitive molecular techniques. This brings me into my analyses, where I use digital droplet PCR, or ddPCR. ddPCR is highly sensitive and can detect one singular copy of DNA in a 1 liter bottle of water. This is very sensitive, much more sensitive than traditional PCR methods, and so it is highly advantageous when studying eDNA and is becoming more of a standard in the field of eDNA. The way that ddPCR works is it partitions a sample into tens of thousands of individual droplets, which means that if we can detect DNA in one of those droplets, we know that we have a positive detection. I had custom primer and probe sets to amplify the cytochrome B gene of my two populations, which is a mitochondrial gene. I developed a ddPCR triplex assay. So the way that this works is I use a combination of different pores that fluoresce at different wavelengths. So I had my two focal frog lineages as well as an internal positive control, which we used to test for inhibition. Using a triplex assay is beneficial because it reduces the amount of DNA extract that we need to use, so it conserves the DNA, and it means that the conditions across the sample are more similar. Now on for some fun results. We found a refined location of this contact zone in southern Ontario. So in yellow here are locations where we detected Pseudacris maculata. In red are locations where we detected Pseudacris triseriata. And in orange are locations where we detected both. Also, this yellow square here is a location from the Laymon et al. 2007 publication, which identified Pseudacris maculata. My colleagues and I tried to sample at that location. However, there is no longer an active wetland at that location because of development, unfortunately, so we weren't able to get a sample there. We identified 10 wetlands where both mitotypes co-occur. This is very exciting because we didn't previously know whether these lineages did share specific habitat, let alone how many there were and where they were. This is not necessarily indicative of hybridization, and my results can't indicate whether hybridization is happening, but it does let us do future studies on hybridization, as some of my colleagues are doing. And now these are results from tissue samples from my colleague Ying Chen, who's a co-author on this work. She could not be here today because she is on her way to Borneo for the World Herpetology Congress, but I'm very excited to share some results that she found. So she found a very similar trend with Pseudochris maculata further north, Pseudochris triceriata further south, and co-occurring populations along the middle of that transect. This gives us some proof of concept for using environmental DNA for this type of analysis because it shows that it is a reliable technique. However, it does show that sometimes eDNA is more beneficial because there were a few locations where I detected both lineages using environmental DNA, whereas tissue samples only revealed one mitochondrial lineage. So this just highlights the power of using eDNA even when we're studying a lineage that is rare. The location of the contact zone is broadly in line with what I would expect from Lockheed et al. 2020. However, I did find much more overlap in the two mitotypes than previously believed. And this is likely due to sampling effort. In the 2020 Lockheed et al. Report, each location is only one frog, so there might have been some diversity that we're missing. We found that this contact zone is quite broad, and there's approximately 60 kilometers in north-south overlap between the two mitotypes. What this could indicate is that if hybridization occurs, there might be weak reinforcement because we know that both mitotypes occur across a relatively wide range, and that competition might also be relatively weak because they're not outcompeting each other. This might not seem like that large of a distance. However, recall that for chorus frogs, this is quite a substantial difference. There are several potential implications for conservation with these results. With these lineages, they are up for reassessment, and so understanding the boundaries of the two mitochondrial populations and understanding how much they overlap is important for designating the populations. So all of this work has potential implications for the assessment and reassessment of these two populations. So in summary, chorus frogs have a very complex evolutionary history, and with our results, we have given some additional information with respect to their evolutionary history. We've refined the location of their contact zone, and we've given some more information not just on the location, but also on the size and shape of their contact zone. We've also found evidence of co-occurring populations, which is very exciting and opens up the door for future studies. We've also developed a method that could potentially be used in future research on these organisms. So using environmental DNA samples with a ddPCR triplex assay, including an internal positive control. All of this work has potential implications for the classification of these organisms and hopefully the protection that they get under the Species at Risk Act. So huge thank you to my collaborators on this work and to other members of my lab. Thank you to the funders of this work who make it possible. Thank you to the chorus frogs for being so interesting. And thank you to the organizers of this conference. And thank you all for listening. Yes?
Hi. I was wondering, so that part of the world has tons of agricultural development. I was wondering if you could talk a little bit about the sites that you sampled. It's like actually kind of hard to find an undisturbed wetland, so I was wondering like where exactly you were sampling.
Yeah, that's a really interesting question. So if anyone in the room didn't hear the question, it was that in this part of the world, there's a lot of agriculture, and so where we found the wetlands. We found a lot of wetlands in roadside ditches. So there's a bit of overflow from a bigger wetland, or there's flooding, or there's rain. And these are all wetlands, or not all, these are mostly wetlands that dry up by late June, early July. Yeah, so it is a very disturbed part of the world, so lots of urban areas.
Yes.
So you mentioned that there may be character displacement at that contact zone. Can you say anything more about what you think is going on there?
Okay, great question. So it was a question about character displacement at the contact zone. So my results don't necessarily indicate that there is or is not character displacement. So I do think that future studies in these locations will be important. In that area, we don't think that there's displacement with respect to calling behavior. So some analyses have shown that in this area, the calls are similar. And sorry, I shouldn't say similar, they're indistinguishable across the mitotypes. I don't want to speculate too much. There might be phenological differences that I didn't capture. I did not detect any landscape differences. So within the areas where I found one mitotype or the other, there's not a difference in habitat. So I don't think it's a habitat difference. So I think that future research on potential microhabitat might be important, but on the scale I analyzed, no habitat differences. So I think that's a very open area for research in these populations in this part of the world. All right, and I think I am almost out of time, so thank you all.
Hi, y'all. Is it time? Hi. My name is Katie. I am mainly a plant systematist and biogeographer. I work at Texas A&M University. I'm a graduate student in the Spalding Lab. And today I want to share with you some work that has been done in collaboration with our lab, as well as several labs at A&M and at the University of Oklahoma. So, for over 30 years, systematists have made significant progress towards developing a molecular phylogeny of all plants. Both the quality and the quantity of this molecular data that underpin the phylogeny continue to improve, and thus so has our understanding of biodiversity, its extent, and the processes that drive its origin. Nonetheless, we're still very far away from the goal of having even half of the known diversity of plants represented by molecular data. In 2015, fewer than 25% of described plant species had any genetic data on GenBank whatsoever. In 2019, data availability increased to about 30%, and then incrementally by 34% in 2021. In our most recent survey of GenBank, we found just over 40% of species with some form of genetic data, though not necessarily phylogenetically useful data. And if we're counting full genomes, less than 1% of species have been sequenced. So as expected, there is a serious bias in this dearth of data. There are taxonomic and geographic gaps in phylogenetically informative data, with most species in the US and Canada being represented. But notably, in the most species-rich areas of the world, huge data gaps remain. Basically, we've made a ton of progress, but the molecular phylogeny of angiosperms is mostly missing, and these missing taxa are geographically structured. So the information content of unsequenced taxa varies across species and depends in part on their probable phylogenetic position. Essentially, not all data gaps are equal, especially if your goal for sequencing taxa is to identify new genes and alleles to understand, what drives diversification or how this has varied across space and through time, or to better understand hybridization, introgression, and how those have contributed to plant evolution. So, in this particular example, in the phylogeny that's on the left, we won't gain a ton of new information about a history of this clade by sequencing species G or H or A or C for that matter. Of course, I'm sure most of us would be happy to have all of the available data for every species, or at least the species that are in our favorite little pet clades. But we all have to make decisions about who we're going to sequence, who is in which species we're going to sequence, and in my own work, these decisions tend to be driven by access and funds, and as a biogeographer, I'm also interested in closing spatial gaps. So maybe there's a more systematic way to go about this to maximize new information gain with new alleles, relationships, etc., so that we would first prioritize taxa that have no data. But we might also like to prioritize unsequenced taxa that have the highest average phylogenetic distance to sequence taxa. So on a small phylogeny such as this one, it's fairly straightforward to see that species D would have the highest priority, followed closely by species E, then I, then J, simply based on the amount of evolutionary history that's unknown in this clade. But on a larger phylogeny, it's not as obvious where the greatest data deficiencies are, so we aim to develop a statistic that might be helpful for quantifying this sequencing priority. Thus, we've proposed a statistic that we're calling seqdef, or sequencing deficiency. This is the equation, which I will quickly talk through. So for any tree with n tips, the seqdef of an unsequenced species I would be related to the average phylogenetic distance to all of the other tips on the tree. Basically, we want to know, on average, how much is known about the evolutionary history in each tip of the tree. This term P is basically a covariance matrix for each tip, so we want to know the proportion of the tree where there is a shared history with the other tips. So if we look at tips B, C, their shared evolutionary history is the branch length between the two purple vertical lines, or 28.5% of the tree. And this is pretty quick to calculate across most phylogenies. Computation time scales with the number of tips on the tree. When we ran in parallel on 16 cores a tree with 1,000 tips, it took less than a minute. A tree with 40,000 tips took a couple of days. So these covariance scores then serve to add weights to the data availability across the phylogeny. The information regarding the evolutionary history of any tip then is simply the average of the weighted data across the tree. So in this example, species C and E have data. For simplicity, I'm going to treat this as a binary have data or don't have data. The seqdef of D would then be the covariance with E times 1, the covariance with C times 1, and then the covariance with B and A times 0, since they both lack data. We average those up, and that is the average information available for tip D. Subtract that from 1, and that gives us the sequencing deficiency, or seqdef. When the data are binary, this scales from 0 to 1. It's not necessary for the data to be binary. Also, to reduce the number of calculations in the current form of seqdef, we're assuming that species with data are not sequencing deficient. Seqdef is dependent on the phylogeny, obviously, and in this case, it's ideally a phylogram. So what I want to talk about briefly is how sequencing deficiency of angiosperms is structured across the phylogeny and across space. I'm also going to compare the sequencing deficiency of rare and common species. I'm using 4 primary data sets in this talk. The first is GenBank. We downloaded the full database and we parsed it by species and sequence type. The phylogeny we're using is from Smith and Brown, which is a synthetic combination of sequence data with Open Tree of Life, so it includes both sequenced and unsequenced taxa. To analyze geographic patterns, we're using the World Checklist of Vascular Plants. And finally, to estimate species rarity, we've relied on GBIF data. We downloaded and cleaned all of the records for angiosperms, and we're classifying a species as rare if they have fewer than 10 species on GBIF. I know that this is super arbitrary and it's super biased spatially, but it's a first start, and we're working on thresholding and quantifying geographic patterns of missing data. So here are some phylogenetic patterns of seqdef. This phylogeny is condensed to show 1 tip per family, colored according to the average seqdef of that family. So red indicates low seqdef, and blue indicates high seqdef. So high seqdef is low data. The 20 most diverse plant orders are labeled. Average seqdef across orders is weakly bimodal with peaks at 10% and 40%, and 23 families have a seqdef over 60%. So overall, sequencing deficiency is lowest in the magnoliids and the monocots, and highest in the asterids, intermediate in the rosids. So the species richness of a family is significantly and positively related to its seek depth. So overall clade richness explains about 40% of the variability in seek depth at multiple taxonomic levels, so genus, family, and order. So it's clearly spatially structured. Most temperate regions of the world are not sequencing deficient compared to tropical regions, and Indonesia and Papua New Guinea have by far the highest seek depth. So we haven't yet completed explanatory models to explain spatial patterns in seek depth, but two correlations are immediately obvious. So first, the seek depth of a region is tied to its species richness. Second, seek depth decreases as you move farther from the equator, with the highest deficiency sitting at about 5 degrees south. So undoubtedly this is coupled with species richness, but we also think that it could be socioeconomic factors as well. So this is the same phylogeny from earlier, but now it's colored according to the average seek depth of only the rare species in that clade. Overall, this figure is much more green and blue and less red than the previous figure, and there aren't any clear patterns as to which clades are better than others. Overall, seek depth of rare plants is nearly 50% higher than in common plants, and we can see that the average seek depth of species decreases with the number of occurrences on GBIF. So our most vulnerable species are, according to this metric, potentially much more information rich than our unsequenced common species. And when we're looking at geographic patterns of seek depth in rare plants, we're seeing that this is a global problem. It's not just a tropical problem. So outside of the United States and Canada, seek depth of rare plants is above 40% almost everywhere. The takeaway here, at least for me, is not that we should go and pull up all of our rare species so that we might glean valuable bits of genetic information, but it is sobering to consider how much data is going to be lost in the biodiversity crisis. So beyond simply identifying sequencing priorities, we think that seek depth might have a number of useful applications. So perhaps it can be a useful argument for requesting funding if, for example, we can standardize deficiency scores across the tree of life. In the same vein, we can calculate the decrease in deficiency resulting from our work in order to demonstrate the impact of our efforts. So this work was supported by CIA Federal Labs, DefenseWorks, the National Science Foundation, USDA, and the Department of Ecology and Conservation Biology at Texas A&M University, and computational resources were provided by the OU Supercomputing Center for Education and Research and the Texas A&M Center for High Performance Research Computing. And that's it. Yeah. Mm-hmm.
I realized that conservation biology is operating something they call evolutionary distinctiveness, which is meant for like pretty similar like purpose. They're trying to identify species that are on long branches, so the ones that we should protect not to lose the evolutionary history.
Mm-hmm.
How much do these two formulas have together?
How's like is it like?
Okay, so like you didn't inspire yourself by the evolutionary distinctiveness. It's like a different.
Mm-hmm. Yeah. Yes, Lydia.
Hi, Katie. Really great talk.
Thank you.
Very charismatic. I love it.
Thank you.
I'm wondering if you could say a little bit about, like, I think, so, like, he's talking about evolutionary distinctiveness, and seek depth obviously tells us, like, how much data we stand to gain for a specific species just if we're looking at pure kind of like genetics and stuff, but are there, like, sort of other features of species or the evolution of species that we might like care about other than just how much evolution on the tree have they done independently? Like, are there other things that we can combine with seek depth to tell us like what lineages might be the most interesting or important to study also?
Yeah, so right now we're working on combining genetic data with trait data for this statistic. So we'll be able to talk about or, investigate species that might have important traits of interest, or species where we don't have any traits but we have a ton of genetic data, stuff like that. So it's definitely a work in progress. Thank you.
right, I'll go ahead and get started. There we go. Thank you guys for all coming to my talk today. My name is Laymon Ball, and I'm a PhD candidate at Louisiana State University. And today I'll be talking about some of the work that I have completed as part of my dissertation. So I study this really cool, really diverse group of plants called Hyliae and the coffee family Rubiaceae. And so this group includes about 29 epiphytic species spread in 3 genera, Hylia, Cosmobuena, and Balmea. And so epiphyte means that these are plants that grow on top of other plants. So in this case, that's usually large trees. And the tribe is distributed throughout Central and South America, but species richness really peaks in Southern Central America. And so species display what we call these hummingbird, hawk moth, and bat pollination syndromes. And so syndrome meaning suites of floral traits that have evolved in response to particular pollinator groups. And so, for example, species that are pollinated by hummingbirds tend to have these orange to red colored flowers that are also tubular and odorless. Whereas hawk moth pollinated species tend to be white and narrowly tubular, night blooming, and they also produce a sweet odor at night. And then bat pollinated flowers tend to be pale green to yellow, also tubular, but more so bell-shaped, and they produce more of a musty odor also at night because bats are also active at night. And so besides that floral diversity, these species also occur in a variety of habitats, but they can mostly be found in low to mid-elevation wet forests. And so this group's distribution throughout Latin America and this high floral diversity raised really interesting questions about how it evolved. And so some of the questions that I've been interested in are, for one, how are these species related to each other? How have geographic distribution, bioclimatic niche, and pollinator use evolved through time? How these variables are partitioned between different Hyliae species? And then what can those macroevolutionary patterns tell us about plant evolution in the neotropics more broadly? And so to first understand the phylogenetic relationships in this group, I had to get some genomic data. And to do that, I decided to design a custom Rubiaceae-specific probe set for target enrichment. And I did that using genomic resources that are all publicly accessible, as well as a few different bioinformatic tools. And I tested the probe set using both simulated and real target enrichment data for 27 taxa across the entire family, which are all included in that tree to the right. And if anyone is interested in the details of how I did this project, please check out this paper published in Applications in Plant Sciences. I've included a QR code there. But briefly, this probe set design worked pretty well, and we were able to get great sequencing data back for the group. And so here is the species tree that I inferred after wrangling all that genomic data that I got with the probe set. So we sampled about 83% of all of the taxa in the tribe and included representatives from all genera and subgenera. 1,375 filtered gene trees went into the species tree analysis, and the local posterior probabilities are shown below the branches. These are all mostly pretty high, meaning above 0.95 throughout most of the tree, which is really great, especially because we did have quite a bit of gene tree discordance in our data set. But most of the phylogenetic uncertainty was coming from this group, Hylia, subgenus Hylia, up there in the right corner. And so to better understand some of the potential sources of that gene tree discordance, I used this program called quartet sampling. And so briefly, this program calculates a few different quartet-based scores, and you can use those to try to better understand whether gene tree discordance in your data set is coming more from just a lack of information or support, or if you might have some evidence of incomplete lineage sorting or another biological process beyond background lineage sorting causing discordance. So the main thing to take away from these results is that we estimated the lowest quartet sampling support values in that group, Hylia subgenus Hylia, and those low scores were consistent with discordance being due to biological processes beyond background lineage sorting, such as, for example, introgression. And I think introgression is a particularly plausible scenario, just because all of those species have that similar hawk moth syndrome morphology. And they all occur throughout the Andes, meaning that there could potentially be some gene flow between those species. But it's still possible that other processes such as heterogeneity and evolutionary rates or base compositions could be causing discordance. And so some of my future work will focus on the phylogeography of this subgenus specifically, and also testing for hybridization and introgression to better understand and tease apart some of these different potential sources of discordance. And so next, I estimated divergence times for the species tree and performed a biogeographic reconstruction. And so the 95% HPD for the node ages and the estimated ancestral states are shown on the nodes. And I estimated that Hylia originated in southern Central America about 19 million years ago. And since then, they've had this really dynamic biogeographic history characterized by frequent dispersal between Central and South America. Altogether, there have been 4 independent dispersals from southern Central America to northern Central America, 5 from southern Central America into the Andes, and then 6 from the Andes into the Amazon. And then once the lineage left southern Central America, we didn't see it return there. So next, I describe species bioclimatic niches using a principal components analysis of environmental rosters from the WorldClim online database. And so before the analysis, I clipped the rosters so that they just covered Hylia's distribution. And so points in this PCA represent land pixels in each of the regions in the map to the left. The first PC axes explain most of the data, about 82%. Or most of the variability in the environmental data, I should say, and they were most strongly associated with elevation and precipitation seasonality. So here we can see that topographic and climatic diversity of the Andes in northern Central America is a lot greater than southern Central America and the Amazon, just based on how much space those regions take up on the PCA. And then to get the species bioclimatic niches from this PCA data, I identified which land pixels in the PCA each species occurs in, and then I took the average score of those pixels on each PC axis, and then used that average as a estimate or measure of niche identity. And so species niche identities are plotted on the right, and they're also color-coded by geographic range. And then from that, we can see that most of the species cluster pretty closely together on that PCA, meaning that they're just using a relatively small subset of all of the environmental space that's available across their range. I also performed an L1OU analysis, which uses Ornstein-Uhlenbeck models to detect evolutionary shifts. And so using those 2 PCA axes as our measures of bioclimatic niche, L1OU detected 2 significant shifts in bioclimatic niche. And so the first was the shift in Balmaeus stormii in yellow as it was dispersing into northern Central America from southern Central America, and also into drier, also higher elevation environments that are more seasonal. L1OU also detected this shift within this Andean subclade of Hylia subgenus Hylia, and this shift represents movement into wetter, higher elevation habitats that are less seasonal. And so we see these 2 significant shifts in bioclimatic niche. For the most part, though, bioclimatic niche has been relatively conserved in this group. And so I think that this might reflect limits that are imposed by Hylia's epiphytic habit. Since these are plants that are getting all or most of their water from the surfaces that they grow on, they probably need to be in environments that receive a lot of consistent rainfall. Balmaea, though, seems to be have been able to broken out of that constraint, and it's the only species that grows in significantly drier environments. And what's also pretty cool is that this is the only species that is variably terrestrial or epiphytic. And so a lot of the time, this plant is actually rooted in the soil rather than growing on top of a tree. And that probably minimizes the water stress that that species is probably experiencing in those more seasonal and drier habitats. And so next, I performed an ancestral state reconstruction of pollination syndromes. And based on these results, we found that Hilly E was likely ancestrally hawk moth pollinated. And there have been 2 independent shifts from hawk moth to bat pollination, one from hawk moth to hummingbird, and then one from bat to hummingbird. And so taking the biogeographic and pollination syndrome reconstruction results together, it seems like most of the pollination syndrome evolution has happened within southern Central America, or sometime during species dispersal out of southern Central America. And surprisingly, no pollinator shifts have occurred in this Andean subclade of Hylia, subgenus Hylia, even though pollinator diversity is really high throughout the region, and there's especially high hummingbird diversity in those more higher elevation environments. And so we have this case where all pollination syndromes are represented in southern Central America, and multiple shifts between syndromes have occurred there, whereas species that have diversified within the Andes primarily have maintained that ancestral hawk moth pollination syndrome. And so I wondered if that disparity in syndrome diversity and the number of shifts that have happened might be related to species ecology, and specifically whether maybe higher competition between species in southern Central America might be leading to more pollinator shifts in that region. So southern Central America is incredibly biodiverse, and in the case of Hilly E, it's the most diverse region for the group. But at the same time, it's also relatively small with much lower and less extensive topography compared to the Andes. And there's also less diversity and less climatic diversity in southern Central America, which we saw based on these PCA results. And so it's possible that species there compete more for pollinator resources just because they're packed more tightly together in that space. And so maybe switching between pollination modes is a way to facilitate coexistence between them. And so to start to get at that question, I calculated pairwise spatial overlap between these species using Schoner's index of co-occurrence. And so my expectation was that if there is more competition among species in southern Central America for pollinators, then those species should co-occur more often than they do in the Andes, and that species with different pollination syndromes should also co-occur more often than species that share the same syndrome. And we see some of those patterns come out. So here are species that occur in southern Central America, and overall pairwise overlap was higher among those pairs. And then species with different pollination syndromes do often overlap, but that isn't always the case. We also saw that hawk moth syndrome species in Hylia subgenus Tetrandri frequently co-occur with each other and with other Hylia E regardless of pollination syndrome. And so one of the reasons why I think that might be happening is because species in this group specifically have really varying corolla or flower lengths, going from as short as 25 millimeters in this Hylia palmata on the left to as long as 75 millimeters in the species to the right. And so that kind of suggests that those species are using hawk moth pollinators with different proboscis lengths. And so even though they share the same syndrome, they're using different pollinators. But I plan on actually testing this and confirming that during upcoming fieldwork. And so just to wrap up, something that I've taken away from this study is that it's not just one factor or another driving Hylia E diversification, but it's really a combination of these geographical factors and ecological ones that are shaping the group's evolution. And it really seems that neotropical topography specifically might play a really important role in shaping the ecological context of flowering plant diversification. And so with that, I'd like to thank all of my lab members. Some of them are here today, and also funding on the bottom. And I went kind of long. Maybe I'll take one question if it's okay with everybody. Thank you so much. Yay, plants! Cool. gotten those questions before, and it's hard to say right now. I have a fieldwork season coming up very soon, and so I'm going to make sure to pay attention to both of those things because I think it does play an important role, especially for the pollinators in terms of like where on the tree like the preference is. So, yeah.
Like the presence of kind of like that sort of like micro niche partitioning, like along
Yeah, right. Totally.
be enough to kind of like cause divergence without driving pollinatorship
Yeah.
in the groups where you see not very much diversity in pollinators. Like
Yeah.
a ton of diversity in where on the tree there are, which trees there are.
Totally.
In the things that have a ton of diversity in pollinators, maybe they're all kind of like still hanging around the same sort of set of trees or something, and so something else is gotta like instigate the reproductive isolation.
Definitely. I think that's a really good point. Yeah. I'll definitely pay